Yeah. Does this work? Over there. Okay, good. Thanks, Heiner. First, I have to apologize because I actually wanted to give a different talk, but my computer didn't start this morning and my whole preparation for the talk last night is gone. So I have to give you an overview talk, but I actually didn't plan to do something different. So essentially, you have to live with that one. I put some slides in there, so there might be some obstacles in there in between, but okay. So what I also changed the title, as you see, relative to what I gave before. But I just wanted to give a little bit of lesson learned in phenotyping from our perspective. And usually I start with the take-home messages first, because people might miss things in between. So the first one is there is not a single phenotyping. There are many of them. And hopefully you will see during the presentation that there are many of them. There are a lot of new technologies, and this is also obviously the link to PhenoRob, mostly non-invasive ones, which in the past, say, decade or so, or probably 15 years now, have very strongly developed and have also changed the whole perspective, because in the end, phenotyping is what a breeder always has done. So this is nothing new, but the way how we do it today is different. But just having these technologies themselves does not guarantee that you have relevant results. So technology is fine, brain first. Make brain great again, or whatever you call it. Integrated R&D approaches are necessary, so you will see that we do a lot of multidisciplinary things in the institute, but also beyond. But even if you have these networks, a single workgroup will not be able to handle this, but there is a lot of interaction necessary. What we often do is what we call phenotyping chain concepts, which integrate between plants from the lab to the field, and also different temporal and spatial scales, because obviously plants are working on the level of cells, but they're also working on the level of globe. So you have the whole range there. Environment from lab to field, again the same thing, plant-environment interaction, and I don't tell you something new here, happens under completely different temporal and spatial scales at the same time, and we need to integrate them. Phenotyping in the end requires the integration of the work into the workflow of the users in order to be applied, and this is a thing which we also have put into PhenoRob as one of the targets, because in the end phenotyping is a tool. We don't do phenotyping for the sake of phenotyping, but for putting it into context of crop management or putting it into context of breeding. I think this is always important to keep this in mind. There needs to be practical solutions, so therefore not only the most sophisticated ones are the ones which will be used in the end. Infrastructure is important, data management is important, usability is important. I'll come back to this at the end. What we often do, and you will also see this in the presentation, we are not only developing these things, but we run infrastructure, and we then give access to other researchers in our own infrastructures, There are a number of really large projects which we are running there at the moment, which we are developing at the moment, where it would also be something which we haven't thought into very deep so far, that PhenoRob, what role PhenoRob plays in that context. Yeah, okay, that's the final statement. Okay, so why phenotyping? Phenotyping is diverse. So if you look into the whole pipeline of breeding, but also in the management part, you see that there are different steps here. And this is a picture which I usually use for introducing that. This is a picture which we use in the Weed Initiative to show what kind of steps are there in order to improve crops. You have a crop design part where you have trait identification. Then you have all the genetic resources which need to be characterized. There is pre-breeding, there is the genetic analysis with large populations, there's the breeding process itself, which has all the selection process, and obviously there is the management part with precision agriculture. All of these different steps require that we get quantitative information about plants and plants in their environment and how they're interacting. So it's very diverse. As you see, we have diversity with respect to what we measure, with respect to how much we have to measure, with respect to how often we have to measure, and if we need to have a spatial and temporal dimension, how this is in there. So it's a very diverse field of activities. In Jülich, we are more or less set up in this kind of pipeline here. This is our institute structure. We are trying to identify novel traits. We have selection and screening of novel traits. We have two groups. We're looking into biomass quality and alternative resources. And there's this whole rim of very important technology infrastructures for us, which are there. So we try to understand what plants are doing. We screen for plants. We integrate it into bigger systems, into production systems. And as I mentioned before, also we are providing access to infrastructures which we have in our place. So this is the people behind there. You know some of them, should know most of them. But what is pretty special with us in the institute is that we have phenotyping, bioinformatics, physiology, molecular biology, and genetics integrated with all these technology aspects of information technology, image analysis, engineering, automation, sensor development. And this is important that we have this in the same institute, because if you do it in the project, just as a kind of reminder for PhenoRob et al., a project is nice, but the likelihood that if you go into conflicts within the project, the likelihood is that people go out of each other, away from each other, and don't fulfill the task. In our case, they meet again next day in the coffee room, and that's very important. So they have to bring their stuff together. So I think it's very important to have this kind of integration going on. So this is a lot of cooperation going there. This is not all our collaborations, but this is some of them. Technology development, breeding and agronomy, crop genetic resources. So I also have to apologize for probably have to leave during the talk of Cyril, because I have to go to the Crop Diversity Trust, where we also have to talk about seed bank phenotyping today, technology platforms and networks. And I'll touch some of them in the meanwhile. This is essentially now running you through the institute from here, novel traits, screening, and biomass, and this is a kind of color code, so the blue parts first. I won't talk too much about roots, because Michelle will do this probably next time in Jülich, because that would make sense. Sorry. So the key approach there is to understand the processes of root structure and function, and to use this to analyze root trait discovery and root trait identification. This is also a thing which I stole from Michelle when she was still in CSIRO, to show how intimate the interaction between the root and the soil actually is. And obviously in many cases what we do in plant physiology, we do things like that. And this is not the same like here. So how do we actually learn from either these things, because sometimes we need to do them, or how can we get actually information over here from this kind of setup. And that's part of the reason why we run a lot of these kind of non-invasive technologies, tomographic technologies. We essentially still get anything from medical which they don't get hold fast enough. So we have MRI, magnetic resonance imaging. We have PET, Positron emission tomography, both systems which you know from medical. So MRI, probably a thing which everybody knows who looks football. There's this thing where people then are put in with their knees and look into where they are damaged. We use this for plants, which makes much more sense. The second one is PET, positron emission tomography. This is also a thing which you know from medical. This is where medical doctors try to identify which part of the brain is active if you listen to talks, or even if you listen to nice songs. So this is essentially based on producing short-lived isotopes, or isotopes which are in the end producing positrons. And then there are these kind of systems here, kind of specific cameras. Don't want to go in detail here with technology, which give us the information where this radioactivity actually is. In our specific case, we use 11C CO2, a short-lived isotope, C11 of 20 minutes half-life. 20 minutes half-life means you can't buy it with Emission, because if you have it, it's gone. So you have to build it yourself. This is the system here, Cypress. This is a small 75-ton machine, which is in a bunker because it's heavily producing radiation. But what we gain is 11C CO2. We feed this to the plant, and then we can see where the radioactivity actually goes to, and where the carbon goes to. We also equip them with robots to get a higher throughput. So there's a system workflow like that. We take C11 from the Cypress, from the accelerator. We put it into a kind of trap to get the CO2, then we load it through a gas exchange system into the plant over here, and then we get the information where this actually works on. So this PhenoPet here is a new system which we are building at the moment together with Philips, because they are giving us their most recent detection systems which have not been used in medical before, because we don't have ethical problems to cut plants and to spoil plants, while this is different with medical doctors, obviously. We have no problem there, so they give us as a kind of early adapters. And what we can do with these things is, oops, that was too fast, things like this one here. So this is, for example, a root system sitting in soil. Oops, here. It's three-dimensional, so this is NMR pictures. And then you can see, and then we apply C11 to the shoot, and then it's transporting down here. This is a distance of roughly 40 centimeters, and down here, for those who sit in the front, you see the speed, the time after application of C11, and you can see how the carbon actually flows in there. And you can also see that this is after 10 minutes, 15 minutes, so it's a rather quick process. So we can actually observe how carbon flow is happening in plants in real time. We can also see that it's inhomogeneous, so this root receives a lot of carbon, while this root here receives little of carbon. And we also can show, or have other examples where we can show that there's a very quick change in the response, how this carbon is actually distributed within minutes. So it's a very responsive dynamic system, and by using these tomographic systems, we can learn a lot about the dynamics of the whole system. Actually, for the first time, we can learn about these kind of dynamic systems. This is another time domain. This is over time, so this is again a root sitting in the soil. And you can see how the root systems is developing, how the side roots are coming up here, so this is day by day. And you can also see that the image analysis in the meantime is really nice, because we can fit them to each other, so the plant, the specimen doesn't sit there for the entire, 20 days, but we have the same species, the same plant and root system in the NMR over and over again, and it fits very nicely. This is also quantitative. On this side you see that the colors here, this is diameters of roots. Again, you can also have the same in this dynamic mode, but this is just to show that it's also a dynamic process. We use this technology to get a lot of information how the plants are actually working. On special structural situation, we can also use it, so nodule development, for example, to look into how nitrogen acquisition can happen. We can also have it as an input for spatiotemporal models. So this would be transport models along root systems here, or specifically in this case it's a sugar beet, and you can get quantitative data out of that. So this is a use where we also have not only nice pictures, which is the first step, but the next step is to get quantitative data. We can do this, and this is, I think, actually things which we did years ago already in CropSense, how carbon is flowing into sugar beets. We can also do, for very high sucrose concentrations, we can do chemical shift imaging, so that we can do non-invasive sucrose determination inside the sugar beet while it's sitting in the soil. So it's a really great tool which we can use in many different directions. Second part, there's something missing here. Yeah, okay. The whole thing of Uwe, I snapped out because he talked about this last week, or last 2 weeks before. So I think this is not in there, so it might come back later on. So I immediately jump. So this just to keep you in mind, there's another layer which is about above ground photosynthesis, crop diversity above ground, canopy structures from single cells to the globe, photosynthesis measurement. So that's Uwe's part, which is also in understanding for crop traits. On this level then, selection and screening, we are developing a lot of technologies. There's a lot of technologies where we have non-invasive methods and standards, where we also have high throughput screening systems. I'll just run you through some of them. A simple one, which is however very important for us at the moment, is having seed phenotyping. We have a robot which is measuring individual seeds for a wide range of different traits. And this robot can measure from Arabidopsis, so this originally would be a dust-like particle. So this is seed of Arabidopsis, to maize or bigger. and it measures for these individual seeds characteristics. This looks like this one here, and it doesn't run, so I have to start it, but you won't see too much. But essentially it's a robot, and this robot picks in an individual seed. In this case, at the moment, does a three-dimensional reconstruction of the seed, and then puts it on the balance. We can also measure individual seeds, like Arabidopsis, for example. That's 5 to 7 micrograms, but we can also go to bigger seeds. So it's picking up a new seed here from this slot here. And then we have different modes of measurement where we can measure individual seeds. Throughput something between 4,000 and 8,000 seeds, depending on what we do. And we also use this thing to measure and to seed, to seed the seed, to put them into the soil afterwards. We also can do this and have done this for plants which actually go into the field later on. And we can also very nicely see that we can actually reduce the statistical error very significantly if we pre-select, pre-categorize the seeds before we bring them to the field, before we bring them into all these expensive phenotyping gears. This is just to show here what this is a very normal, I think this is barley, where we are going in a volume or a mass between 1 milligram and 3 milligrams. So this is a normal diversity of a seed lot. And you can easily imagine if you have a seed which has 1 milligram, this is different from a seed which has 3 milligrams. So what this robot also allows us to just cut out this one individual band here, and then we have much less statistical error in our later on analysis. This is an ideal situation that you have this Gaussian distribution, but you can also have, and in many cases we have multi-peak situations, and just characterizing the seeds before you bring them into the field is something which is very useful in many cases. There's a lot of applications here which I don't want to go into detail, so analyze the seed population itself, so after harvest, or reducing the statistical noise, or doing seed physiology, or what I will talk to the guys later on in crop diversity trust about seed bank phenotyping. That's an option which is there. We also use it to use seed to plant tracking. So for each plant which we put into our equipment, we know exactly what the seed quality, the seed structures are. And then all this data which goes along the lifetime of the plant is going into a database. So that we have a full life history of each individual harvested plant, which we have in our measurement systems in the end. So this is things which we often do in greenhouses, but we also go into this kind of phenotyping chains from single plants. But we also know that single plants are just one system approach, but in the field we have a multiplication or a kind of cluster or assembly of plants. So we go to blocks of plants, several blocks together in the field, or then into Klein-Altendorf, as for example, as a complete field site. We also do meta-analysis. That might also be a thing where we could go into a little bit deeper at some stage to how to utilize all this different information which is already in the publication databases to identify phenotypes and real background knowledge about phenotypes. We do this for a lot of growth, biomass and shoot structure. We have a lot of photosynthesis methods, and Uwe has definitely told last time a lot about them. We have water relations. We do active and passive thermography. Not sure if this was in there as well, but this is kind of thermography, using thermography to get quantitative information about plants. We also do quite some work on microwaves, because microwaves are a quite nice approach to get information about specifically water. And then the next step is obviously to integrate all of this into one system, into an automated system for higher throughput. We have a number of different 3D systems. This is another robot which is now, I should actually exchange that to the real picture, which we are now having in place. This is a robot which has a possibility to get a large number of different, in our case, in this case here, 3D measurement systems, and the plant would be sitting here. So this is not the bottle, this is a plant theoretical surface, let's put it that way. And the robot essentially goes around it in different directions, pretty high speed. Therefore, this is in a whole massive garage in here. and then we can mount many different sensors on that. So this is very good for auto calibrating and getting information what kind of sensors are best used. And you get all this kind of different measurement modes here. In this specific case, it's for 3D measurements, and there are a wide range of different sensors which we can test there at the same time. And the same sensors would also be, all of them would be mounted on the same robot so that we can actually measure different things here. As I said, then it goes into kind of higher throughput, either in growth chambers, oops, wrong direction, growth chambers under controlled conditions where we have a number of robots which are running in there. Also in greenhouses, this is speed up, so don't think this is going that fast. This is actually in Klein-Altendorf. This is more on the assemblies in container-based systems. Looking to Uwe, you talked about this, okay. There are also, so there's a number of technologies, and just to keep in mind, all these kind of systems we build ourselves because we have our automation engineers, and this is, for us, it's very important to have them in place, and also the sensor people. This was also shown by Uwe before, so that's the reason why I put it in, just to remind people. And here it says, Salmina Uwe Rascher. So just relate back to what Uwe said 2 weeks ago. We also go with the screening system below ground. Here we have systems which we essentially go into risotrons. Risotrons are kind of some box in that specific one, which I will show in a minute. It's 70 centimeters wide, 1 meter deep box where one part is transparent, so that if you incline it and the root grows along the transparent screen, you can watch the plant, how it's actually, the root, how it's actually growing. And we do this again a little bit more automatic. So these systems here, this is the risotrons sitting here. This is our imaging box, which runs along these things, and they are drawn then into the box. Then the whole thing closes because we want to have most homogeneous imaging conditions. And based on that, we then do 2.5D reconstruction of the shoot because we take pictures from different directions. And we also take pictures from the root system, which looks like this. And because we can do this repeatedly, we can come up with something like this. So this is the same barley specimen over a period of whatever, 12, 15 days. And you can see how the root system is developing here, how the side roots are coming here. So this is to say a little bit similar to information like in the tomography system, but it's much less, it's much more invasive because we obviously force it into 2D plane, but we can link these kind of information to the tomographic systems. And then we get quite a number of different traits, above ground, below ground, which we are putting into the databases and learning about genotypes, and also be able to put some different environmental conditions down here to see how plants are actually responding to that. This system here, which you can't see now anymore because it's black again, so the system which you saw in the video before, this has a capacity of 72 risotrons. We have a second generation which has 250 risotrons, and we are just about building the big one, which is this one. This is a new greenhouse which we finished some time ago. This is one compartment of this. This is a 800 square meter compartment where we are now presently setting up all these risotron modules here. This will be 1,500 to 2,000 risotrons, and at the moment we're building these first 5 imaging stations. There might be more. But in the end, we should have a real large number of risotrons to measure there. The whole thing is run based on these kind of, you probably can't see it from the back. This is our autonomous running robots from logistics. They are taking modules of risotrons, transporting them to the imaging stations. They are automatically unloaded, and in the meantime, the robot goes somewhere else. So it's a really logistics center. We have 5 of them, which will be running until end of the year in the greenhouse. Or hopefully they will run end of the year, and then they will be running much more longer. So this is a system which is presently in the development or in the setup. If you go in the greenhouse, or if you come in January, we should be able to run that thing. We have to be able, because we have to finance it until the end of the year. Then there's the next step, going into the field. Again, probably Michelle will talk about this some stage later. Therefore, I moved over to not the classical crops here, but to show you what we do there, not only in Jülich or in Germany, but beyond. That's a cassava store project where we work a lot with our Thai colleagues, also on the tech side. So there are biotech in there, there's NECTech, which is the engineers in there. And we are building there an experiment, or have an experiment there, where we are running 600 cassava lines in Thailand. We are sequencing them, not only the 25 initial genotypes now, but also the 600 genotypes to achieve us on that. And there's a lot of storage root development measurements in there. There's phenotypic and genetic variation in there. And the aim is to increase the yearly production, obviously. We do that in the field. So this is the field in Thailand, where we have a lot of measurements installed there at the same time. And this is, again, the field in Thailand. And the important thing is that all this data, which we have in the field here, which are generated there, and you see that's a lot of data, that they are transported from, this is Rayong, so it's some 200 kilometers away from Bangkok. They are transported to Bangkok and to here, and then we mirror them each day between Jülich and Bangkok, so that we have all the same data available. So this is a little bit on a bigger scale, the Klein-Altendorf-Jülich situation. So how do we transfer data? That's working with a pretty large number of data, so we have some experience in that context, and probably will also bring this into that context. As what kind of data do we get there? This is shuffleomics on cassava roots. As you see, this is different weeks, how these different genotypes here are developing, how the storage roots are growing. This is image. We take then images in this box. You see a little bit of a cassava root in here. This is turning. Videos are taken, and then we get quantitative information about these cassava roots to store them. And then we obviously get quantitative information about these root systems in bringing that into the whole context. So this is a little bit into the direction of what we do at the moment in the fields in that context. As I said, the outside Germany context, just showing that we do also a lot of activities in this direction. There's a full new part, or it's not too new anymore. A part where we are also working on is not the high-end phenotyping, but the low-end phenotyping, let's put it that way, but it's much more than what people could do before. So how to enable smartphones or laptops with new technologies. There's an awful lot of sensors in there. Just last night I got a laser measuring system for an iPhone, which is really great, where you can measure a lot of things with. A lot of plug-on sensors, thermographic systems, and then obviously quantification, or the app part needs to be built on that. We also have released some of these things already, but this is a complete domain which is presently taking up in the phenotyping community quite a lot. Activities where we have this kind of low-cost phenotyping, which obviously is very interesting also to put them into the workflow of the breeder, but on the other hand also into more developing countries. There's a lot of things actually going on there also with our Thai colleagues, but also globally. As I said, there's this whole cooperation part in there, and this brings me to the last part. I had to speed up because I only was allowed to talk 30 minutes, so no, it's okay. As I said at the beginning, phenotyping as we are running it is a lot of interactions and networking. So this is a selection of some of the networks which we have run or do run or have been included in the last years. So Jülich Plant Phenotyping Center is kind of our small scale technology development and implementation part in the institute. CropSense is very well known here in Bonn, where we had this interaction on the cluster, complex agronomic trait, these characteristics. This was a lot of technology development, but also implementation. Then we had, or still have, until end of the year, the German Plant Phenotyping Network. This is a project which was mainly aiming, or only aiming, for the update and installation of infrastructure and technologies. So I come back to this in a minute. Then we have the EPPN and EPPN 2020. This is mainly projects coming from the European Commission to allow access to infrastructure technology, so infrastructure installations in another country. So this is, and I think I'll have a slide on that as well. At present, we are developing a pan-European plant phenotyping network, the so-called EMPhaSIS, or research infrastructure. That's an S3 infrastructure. I also will give a few things on that. And then we have the IPPN, the Global Phenotyping Network, where we are collecting all the globally, most of the actively phenotyping institutes into that kind of community. That's a different thing again. And then data integration standardization. This is also part of the reason why we have all these networks, because obviously standardization is key if you want to exchange data. And therefore we have pretty strong activities there at the moment on the global side. We are also together with the third director in the Institute, Björn Usadel. He is very strongly involved in the minimal, in the data standard part, and this is MIAP as one of a joint activity between the EMPhaSIS and Elixir, which is the bioinformatics S3, where we work together and identify what kind of minimal data set is required in order to deliver useful data to these databases. Just some short information there about the DPPN. The DPPN is the German Plant Phenotyping Network. It's a national infrastructure, 34 million euros for 6 years. In Jülich, Gadersleben and Munich, essentially building on existing phenotyping competences. I showed already a little bit here. Gadersleben is a Leibniz institute. They had a lot of information, a lot of background in LemmaTech platforms, so commercial platforms. They also have a lot of activities in the gene banks. Munich was very strong, or is very strong already in environmental simulation. And we use that in building this phenotyping, because phenotyping is a tool, into the research background. In our case, mainly root, shoot, lab, field, resource use efficiency. In Gadersleben, they have the gene bank and a lot of omics technologies, and Munich does the plant pathology and plant microbe interaction in that context. So this was the reason why these three groups were actually selected, plus, and that's the thing which is not on that slide, these three institutions agreed to run these facilities for at least another 10 years after finishing of the project. And that's a thing which we can only do if we have basic funding, and that's now starting into next year. What is done in DPPN? We have a number of modules. We'll have root architecture and physiology. We have shoot architecture and physiology. We measure field, stand architecture and stand physiology. And we have the patho parts. These are formed together, and I showed you the large-scale rhizotron systems. There we also measure the shoot part. There are extensions of the LemmaTech systems in the field. There's a lot of field phenotyping, again, showing back to what also Uwe said before. I guess you also talked about breed phase. That's also an installation coming out of DPPN. And there's a lot of patho parts, which is more in-house and more specific things, which we are not too much involved in that. This is the outline of the project. So you see it's quite complicated. With all the different modules which we have in there. We are by far not for PhenoRob in that kind of detail yet. We probably should not go in that kind of detail and be a bit more flexible, but because in this case it was about setting up defined infrastructure, we had to have a very stringent management there. This is not only happening here in Germany, but there are a number of other national infrastructures. The other one which has a similar size is Phenome in France. All the others are kind of more or less coming up now. This is national, so Germany and France were more or less the early runners. Australians was a little bit before, but they have a different structure. So these are the three major ones. Australia is here, sorry. All the others came up within the last 2 years. So there's a really large development, strong development going on there at the moment, providing these national platforms. They have different nature. There is a lot of investment there. If you just to put you a little bit into perspective there, on the European side, in the last 5 to 6 years, there was and now with the new ones, there's an investment of roughly 350 million, which was invested in Europe. CPPN, the Chinese plant phenotyping networks, at the moment they are negotiating on 2 projects, each of them 300 million. So that's the kind of dimensions which we are handling there. This is also clearly the indication that we have to work together, because we will not be able to handle, to act as a whatever, even DPPN individually relative to a 600 million investment in China. No way. So how do we do that? We build this kind of regional clusters. That's also the reason for emphasis. That's also the reason for all these EPPN things. The North Americans have come together. There's another investment of the same dimension like in the Chinese. So there's a lot of regional networks going on, and it's clearly a field of activities where there's a lot of activities going on at the moment. And therefore there's also this kind of crop-specific aspects, like for example the wheat initiative. But we have then formed the IPPN, the International Plant Phenotyping Network, as a international coordination body or membership-based body which tries to somehow organize these things globally in order to avoid specifically that people always do the same kind of mistakes, because that's the thing which I think we are horribly bad in as scientists as a whole. We only describe our success stories, but it would be more important to show what is not working, because other people will work on that anyway. So if you have 10 solutions or 10 options, then probably 9 out of 10 will not work. And if you only publish the one which is working, then I can guarantee you that many other people are working on the other 9, which you already tested that it won't work. So this is a lot of failed effort which we somehow should come together to avoid that. On a European scale, as I said, we are developing emphasis with this nice acronym. We need that acronym because we have this long title: European Infrastructure for Multi-Scale Plant Phenotyping and Simulation for Food Security in a Changing Climate. This is program, this is acronym, so emphasis is working on that. We are building emphasis as an S3 project, so a European research infrastructure with having basic synergies, innovation parts in there, and access. I won't go into detail there for time reasons. This is an ongoing project where we develop an integrated pan-European infrastructure, where we also have the data information systems which are aligned between all these different infrastructures. And there's also development, evaluation, knowledge and technology part in there, where we then talk with users, talk with companies in order to spin out technologies. We are on the roadmap, S3 roadmap, since 2016. The roadmaps here, this is the projects there are not handed in by scientists but by the governments, because in the end the governments are deciding what's going on there. We are now running the prep phase project, which is a European funded project, but the aim is to come up with a business plan for emphasis, and then hopefully from 2021 onwards we will have a legal entity for emphasis, which will be a European legal entity, a European research infrastructure, which will run all these kind of processes and services for the community, and then we hopefully at some stage are an emphasis landmark. What do we do in emphasis? There are 5 different layers. One is about deep phenotyping platforms, so a lot of the stuff which I showed you before, in-house tomographic systems, controlled environments. This is in this one, deep phenotyping. A second one is intensive field platforms. And here, obviously, the activities which we do in Klein-Altendorf or the activities which we do or plan to do in PhenoRob would be very nicely fitting in here. Then we have lean phenotyping, as again a thing where I think if we really want to talk to breeders or farmers in the end, we need to talk about this one, which is much closer to the real life of a farmer and the breeder. So how do we do that? Then there's the modeling platforms, because even if we have a lot of machines, we will not be able to measure all of these things. We have to model, and we have to integrate between the phenotyping and the modeling. And then we have the whole joint data management and e-infrastructure, which is a very strong activity at the moment, which we specifically through IPPN and emphasis roll out not only in Europe, but also globally to get agreement on what kind of data management and e-infrastructure should be there. Just to give you a short, there's a little bit feedback on what kind of facilities are in there. These are the high resolution, high throughput systems. I showed you this one, that one. This is a high throughput CT in Nottingham. This is a system in Ghent. So there are also some of these systems in other places. It's by far not complete. Then we have the semi-control systems. I have to kick out that one because this is a real old one. So this would, for example, include Klein-Altendorf activities. This is the kind of automated phenotyping robots. There's a lot of experimental systems out there. Networks for field sites for practical experiments, where we could also think about mobile systems. And again, here PhenoRob might be an important thing where we can also hand out at some stage machines and sensors to breeders and farmers. Then we have the modeling platform, mainly into disentangling complex traits, doing genetic analysis. This one goes more in the direction of another S3 project where we are collaborating with. And I showed you this before. This is we are presently running a database for infrastructures. At the moment we are, I think it's now over 180 infrastructures which we're having in that platform, which we put into the database as well, which people can search then for certain parameters which they would like to measure. And the last one, IPPN. IPPN is a membership-based organization. We are now, it's not in here, since yesterday I think we are 38 research organizations, which is strongly growing now. We also have opened for industry now. This is trying to integrate globally the fragmented activities and enable exchange and also knowledge exchange and people exchange. We have a number of instruments, symposia, work groups, workshops and meetings and interactive communication platforms. And yeah, this is essentially was the last one. I could talk more about that. But take-home messages again. I hope that I could show you that phenotyping is not a single entity. There are many. Technologies are important, but as I mentioned before, you need to use your brain to make something useful out of it. R&D, I showed you that we do quite some diversity in different disciplines, imaging, imaging, computing sciences, omics, bioinformatics, automation sciences, and plants. Then phenotyping chains, I showed you a little bit lab to field and for plants and environment. It's a tool in the end, so we have to build things that actually works into the workflow, so there the visualization activities come in very strongly, and yeah, that's the last one again. So hope that there is something in there for people in PhenoRob, and that we can extend the activities which we have so far into more robotics and data sciences. Thanks very much.
Thank you. Other questions?
Uli, what are your efforts in terms of the kind of mobile phone or, let's say, lightweight mobile sensing phenotyping area? Because that's something that could be also relevant, because it would allow us and all the teams in PhenoRob to actually acquire data on a larger scale. Everyone could have those devices, and you probably make progress faster with a lot of those small devices. At least that's my feeling.
So
what's the status there?
We have a work group of that in IPPN. We do some work in Jülich. For example, there are these kind of plug-on thermographic systems. Interesting technology development because it's very quick, and we have done some work on that some time ago where we compared industry cameras versus the plug-on ones. The plug-on ones are the same quality, industry 12,000 euros, plug-on 230 euros, so it's a really game changer with respect to cost. There is a large number of different sensors coming up. As I said last night, I got a small package with a laser scanner on a smartphone. It works quite nicely. I think it's I think it would really be something to think about it more in depth, because it would be something where we can translate the knowledge which we have in all of these kind of different things more closer to the end user. Not even talking about citizen science, because that would also be possible. There's a lot of sensors already today in the smartphones. We have all these kind of double camera systems to get three-dimensional pictures. That's already there. I don't think we use it properly yet. But there would be, I think this is something to think really in depth, because in the end, if it will again only work if you bring it into the reality in the day-to-day life of people, so that we have in the end apps which people can use. And that's the thing where we at the moment Jülich have few of them, very few of them, but I think that's the thing which we still need to think more. So it's a sensor part, it's a rapid development which runs anyway, and where we need to think properly where we put effort in.
Thanks. So I also understand that the sensor technology which is there is surprisingly good compared to the price if you actually can do something reasonable with it.
Yeah. Yeah.
Thanks.
I guess just to comment on that discussion, I mean, the other aspect of mobile phones that we're discussing is now you can attach a microscope type lens and system onto them. So for the sort of mechanistic or scientific or trait discovery aspect, you can have your app collecting your macro data, and then if you want to relate that to the details at the cellular level for diseases or the rhizosphere or whatever, I think this is really exciting too. And we just, I think we need to follow. We're not.
And we are by far, we are far behind others. So if you talk about NSTDA, so they have done this kind of microscopic things. They send it out to schools. They have devised things where there are 3 different resolutions. They send it out to schools. This is cents of cost, and that's just a service which they provide. They don't talk about it. It's just do it. The same also on not on the school side. So this is also a dissemination part, but on the other side with respect to pathogens, for example. Again, Thailand, they have a network where they take images. From a farmer takes images, it sends them to an expert. The expert looks on it and says, this is the kind of whatever spray, this and that, and it's a kind of extension service. They do the same with nutrients, so they get kind of color code, put it next to the plant, they measure it, and then they put it onto the expert still. But in the future, this will be artificial intelligence, telling them what kind of nutrients they need. So they're already there, but we are just talking about it.
Okay, further questions? Yes, Rihanna.
You have said that one take-home method technologies and does not guarantee relevance as any data analysts where there's stuff and so the first question, the second one, how far do you think to the standardization of the readouts or what to aim in the different technologies?
I don't want to. I would say we can probably go into many publications in the last 20 years and see that. That there's a lot of, so there was a period where there was just, people were just fascinated by technology and they measured everything, and in the end, what is the output? Not really something. So the only way how we can do that is really integrate early on with the users. So that's the kind of what I mentioned with this kind of get into the workflow of the users. We talk very intensively at the moment with breeders. There will be a workshop financed by the Ministry of Agriculture end of early next month, where we specifically target what are the hurdles that we have the technology, but they are not taken up in breeders, and there are many of them. The reason why I'm going to the crop trust now is I would like to bring phenotyping, whatever it is, even simple things, into the workflow of the seed banks. They are sitting there with millions and millions and millions of these small bags, and they have no clue what's in there. And the moment what is happening and where they're spending a massive amount of money is for sequencing all of them. So, but phenotypic information is missing. So, okay, then you have a nice sequence. So what a breeder in the end can deduce something from the sequence, but they don't have any phenotyping data for that. So a lot of the discussions there is we really have to talk with those people who are using these things, and this is not only academics, this is farmers, this is breeders, this is seed banks, this is horticulture, you name it. So the whole thing, I think I had it in there at the starting, also in the take-home messages. A lot of this rapid development which we did in the last years was because all of technology comes available. But it's not enough. We really need to put this into a proper background. I can give you 100 examples where things failed, but no.
And the second one, the standardization process, how far
So companies do that already. Companies are quite okay with standards, but they're islands. So think about the merger of Monsanto and Bayer at the moment. One of the biggest problems they have is different standards, and they are not fitting to each other, and they are reaching back 25 years because they didn't think about it before. So there is a lot of standards already out there. Standards need to be a community effort. Therefore, doesn't make sense that we do it in Jülich. Therefore, we do it already on a European scale. And feeding that standardization, so what we do in emphasis is fed back into NSF, into the Australian community. There are links there. Into the Asian community, there are links there, so that we try to build a kind of global perspective of how this should be looking like, and also the infrastructures for that. So we don't want to, and that's probably the way to do it today. We don't want to have all the data in the one database, but rather reach out and keep the data there. But at the same time, we need to make sure that the data there are good data. So crap in, crap out. So we really would like to set up high quality standards for phenotyping. And that's what we also implement, for example, through this EPPN 2020, where we have certain levels of qualifications. Emphasis will also have a quality assurance level in there, so that you can only get, for example, this kind of transnational access projects if you can guarantee a certain quality of data. Otherwise, forget it. We will not let these infrastructures in. So it's a kind of pressure thing. We also talk with publishers. Publishers will, in the future, hopefully only accept certain standards. At the moment, this is a mess. But at the same time, these standards need to be responsive to technology development and to the development as a whole. So as usual, standardization is a process rather than a fixed thing at one point in time. And we also have therefore talked very intensively to funding agencies. Actually, they started with that already some years ago on AeroNets, where they said we don't want to have that situation that we always fund this kind of small projects, and later on people say, sorry, I have no idea where the data are because the PhD student is gone. So we have this, there's the knowledge, there's the data management included in there already. So funding agencies asking, so if you want to have that project, you have to have a data manager and you have to have a data management plan. And the data management plan in the next step will have to follow certain rules which are set by the community, and therefore the funders at the moment and the two part-time other ministries are building up these kind of networks of communities which will then come up with a certain standardization process. So for example, MIAP, there is a MIAP 1.2 standard at the moment. There will be soon a MIAP 2.0 standard. And we expect that all the projects which are running EPPN or EPPN 20 projects from the Commission, that they apply that standard. Otherwise, they will not get money.
So, next question.
Okay, well, I am most interested if it would be possible, in addition to the carbon transport in plants, to visualize also the nutrient uptake or microbiome uptake, and but also maybe the transportation within the plant. So is it already possible?
So carbon is the easy one,
Yeah.
which we have established because C11 is there and we have C11 there. If you want to do the same with other elements, let's put it that way, it very much depends on what kind of element it is. Nitrogen, for example, there is a nitrogen element which can be used, but it has a half time of 6.5 minutes. So that puts another layer of urgency on how quickly you handle your experiments. So C11, after 3 hours, the radioactivity is gone, so you can only do experiments within these 3 hours, 3-hour windows, but then you can do another one. So it's, in principle, it's possible. But with 6.5 minutes, you're running already pretty strong. The cyclotron itself can do nitrogen. We've done that some years ago. And then with nitrogen, there's another problem in there because you need to put nitrogen in the right form. So if you want to have nitrogen into ammonia or nitrate, you have to make sure that this is really put in the right form. So on top of the short lifetime, you have to do the kind of separation or put it in the right context. If you want to have it in amino acids, I did that during my diploma work some centuries ago, last century. We essentially applied C11 to plants and did the separation on columns, and then we had relatively pure amino acids labeled with C11. That's possible, but it takes time. If you go into other elements, it depends on the availability of a short-lived isotope, which is a positron. There are quite some, but many of them have these kind of very short lifetimes, and essentially we also have a group in Jülich who is doing this also for the medical applications. We talk to them since, I'm in Jülich now since 18 years. I talked to them since, say, 15 years that we would like to have certain elements. They always say, yes, that's possible. If you give me a postdoc for 3 years, we can do that. Because just making them is an issue. Yeah? Because a lot of the stuff which they do there is working on Märklin-Eisenbahn. No? So you have to fully automate the full chemistry. Yeah? So they have these small trains running from one beaker to another beaker, keep putting it. So it looks nice, but it's real work. No? But in principle, there's heavy metals. There are many things out there which could be done.
Okay. Thanks again.
Yep.